Wow, what a fantastic and insightful question. I want to thank a viewer, and you know who you are, for bringing up such a thought-provoking challenge that really digs into the very core of evolutionary biology, the philosophy of science, and the complex world of biological nanomachines. Your detailed breakdown gave us a great way to give a full answer. Part 1. Do all these mechanisms lead to universal common ancestry? Of course, the mechanisms of evolution, natural selection, genetic drift, mutation, and gene flow are not only consistent with universal common ancestry, or UCA, but are the forces that, through their operation over vast periods of time, lead to it. The concept of UCA is not just a hypothesis. It's a fundamental pillar of modern biology, supported by an overwhelming confluence of evidence from diverse scientific disciplines. Let's look at the following evidence that strongly supports universal common ancestry, genetic universality. The strongest proof is in our shared genetic code. The fact that almost all life on Earth uses the same 4 building blocks for DNA, A, T, C, and G, and the same codes to make the same proteins, is super powerful. If life started many times on its own, it's highly unlikely that each time it would end up with the exact same genetic language. Also, finding similar genes that come from a shared ancestor, in very different species, like the CHOX genes that control body plans in everything from bugs to humans, shows a clear genetic link back to a common ancestor. Beyond genetics, life's basic chemical machinery is, well, remarkably similar across all living things. Things like glycolysis, which is how we break down sugar for energy, the Krebs cycle, and how L-amino acids are used in proteins all point to a shared chemical history. The double-layer structure of cell membranes is another universal feature, and honestly, it's really hard to explain them coming from different origins. Now, if we look at the fossil record, even though it's not complete, it consistently shows a pattern of life, evolving from simpler to more complex forms over long periods of time. Finding fossils that show these in-between stages helps fill in the gaps in evolution, showing those gradual changes that connect different groups of organisms to their common ancestors. The amazing similarities in the bone structures of vertebrate limbs, like the 5-fingered limb, even though they do different things, for example, a bat's wing, a whale's flipper, or a human's hand, are best explained as changes from a common ancestor structure. Similarly, the likenesses we see in the early development of different species embryos really show their shared evolutionary past. When it comes to how different species are related, the ability to make consistent family trees, those diagrams that show how living things are related, using different types of information, whether it's from genes, body shapes, or chemicals that all come together into one clear tree of life, gives strong support for all life coming from a single common ancestor. And honestly, the more proof we gather, the more detailed and connected this tree becomes. Given the deep, fundamental similarities across all known life forms at the molecular, cellular, and structural levels, the most logical and, simplest explanation is that these shared features were inherited from a single common ancestor. To propose multiple independent origins of life, each somehow developing practically identical fundamental systems, really stretches belief and goes against the principle of parsimony. The simplest explanation is usually the best. From a philosophical perspective, Universal Common Ancestry provides an elegant, unifying framework for understanding the biological world. It transforms life's seemingly disparate diversity into a single, connected narrative of descent with modification. While absolute proof, in the strict mathematical sense, is hard to come by for historical scientific claims, the overwhelming and growing body of conclusive evidence makes Universal Common Ancestry an empirically sound and, well, practically undeniable scientific conclusion. So, we move on to the second part of our discussion, asking ourselves, can naturalistic evolution really explain the existence of purpose-driven nanotech machines that operate within an integrated information system? This is, honestly, a pretty complex and often debated topic, and it often touches on arguments about irreducible complexity and intelligent design. But, from a naturalistic evolutionary point of view, the answer is a resounding yes. These intricate biological nanomachines and integrated systems can be explained by gradual evolutionary processes. Now, let's clear up what we mean by these terms in biology. Purpose-driven nanotech machines refers to complex molecules like ATP synthase, which makes energy for the cell, ribosomes that build proteins, bacterial flagella for movement, and various molecular motors like kinesins and dyneins that carry things around inside cells. These are, without a doubt, highly advanced. They work at a tiny scale and do specific jobs or tasks that are super important for life. When we say integrated information system, we're talking about the cell itself, a highly organized network where DNA, RNA, proteins, and signaling pathways all work together smoothly. The information here is stored in the genetic material and is processed through these complex control networks. So, how does naturalistic evolution explain all of this? Well, evolutionary theory suggests that these complex systems came about through a series of small, step-by-step changes, where each step along the way was helpful in some way. Here's how it works. Exaptation, or co-option, is a really important idea here. Think of it this way. Existing proteins, or parts of proteins, which might have done one job originally, can be used for a brand new and totally different job as part of a bigger developing system. A classic example of this is the bacterial flagellum. Many of its parts are similar to proteins found in the type 3 secretion system, or T3SS, which bacteria use to inject proteins into host cells. This suggests that parts of the flagellum likely grew out of an existing T3SS, which already had a useful purpose. Gene duplication is a major source of raw material for evolution. When a gene duplicates, one copy can keep doing its original, important job, while the other copy is, free to gather changes without immediately hurting the organism. Over time, this duplicated gene can change, getting new functions or becoming special to work with other proteins, which could lead to new molecular machines. Molecular machines are often made of parts. Evolution can build these machines by slowly putting together individual parts, each of which might have had a simpler, basic job on its own. Simpler, less effective systems can act like a framework that helps more complex ones develop. Even if the original framework is lost or changed a lot later, it played a very important role in how things evolved. The first versions of these machines didn't need to be perfect. Natural selection favors any small improvement in how something works that gives a survival or reproduction advantage. Over very long periods of time, these small improvements add up, leading to the very efficient and complex systems we see today. In some cases, complex structures and functions can come from simpler parts through things putting themselves together, guided by what the molecules themselves are like, often affected by the environment and what natural selection chooses. The information in biological systems is stored in the DNA and RNA sequences. The integration comes from the complex regulatory networks and feedback loops that control gene expression, protein making, and cell communication. Evolution explains this by the slow buildup of helpful changes in both the coding parts, which leads to new proteins or change protein jobs, and the control parts, which leads to smarter control and teamwork. Natural selection is always sifting through random changes, keeping the ones that make the system better overall. Now, people who believe in intelligent design, or ID, say that some biological systems are irreducibly complex. This means they can't work if even one part is taken away. And so, they couldn't have come about through slow, step-by-step evolution. The bacterial flagellum is often given as the main example. Evolutionary biologists are actively addressing these claims by suggesting believable step-by-step paths, often involving exaptation, where parts had different jobs before they were put together into the complex system we see now. The ongoing challenge for evolutionary research is to figure out the exact historical paths for all these complex systems, but the general ideas and many specific examples show that it's possible. From a scientific and natural point of view, the purpose of these machines isn't a smart, planned design by some intelligent being. Instead, their purpose is simply the job they do, that natural selection favored because it helped the organism survive and have babies. It's a way of describing the outcome, not a sign that a designer had a specific plan. So, let's talk for a moment about the information debate. While biological systems undeniably contain and process information, the origin of this information within a naturalistic framework is explained by the accumulation of beneficial mutations and the filtering effect of natural selection over vast periods of time. There's no need to bring in an outside smart source for this information. In fact, it comes about naturally through evolutionary processes. Now, moving on to the big picture. Universal Common Ancestry. Yes, based on the huge amount of scientific proof from genetics, biochemistry, the fossil record, comparative anatomy, and phylogenetics, the ways natural evolution works strongly point to and completely agree with universal common ancestry. This is, honestly, a main part of modern biology. When it comes to nanotech machines with a purpose and built-in information systems, naturalistic evolution can explain those too. Even though the complexity is huge, scientific research is always finding possible evolutionary paths that involve things like exaptation, gene duplication, gradual assembly, and picking out small improvements. The purpose here is about how it works, not about someone's intention, and the information comes from random changes filtered by the sure process of natural selection. This area is still a big field of active research, and yes, even though all the steps aren't perfectly detailed for every molecular machine, the general ideas and many specific examples show how such complexity could come about without needing explanations that aren't natural? Your question really shows how deep and powerful the theory of evolution is. It's a field that's always taking in new discoveries and making our understanding of life's amazing complexity better. Is there a specific nanotech machine with a purpose that you'd like to look at more closely regarding its possible evolutionary path? You're absolutely right to point out the incredible variety of purpose-driven nanotech machines within biological systems. Life is literally full of these tiny molecular wonders, and each one does very specific and important jobs. They show us clever ways to solve problems, like changing energy, moving things around, handling information, and keeping things strong at a very small level. So, here's a breakdown of the different kinds of biological nanomachines, grouped by what they mainly do, with some examples. First, let's talk about molecular motors. These are all about movement and transport. These machines turn chemical energy, usually from ATP hydrolysis, into mechanical work. This lets things move around inside the cell, or even the cell itself. Take kinesins and dyneins, for example. Their main job is to move things like vesicles, organelles, and chromosomes around inside the cell, along microtubule tracks. Kinesins usually move towards the plus end of microtubules, while dyneins move towards the minus end. You can think of them like tiny delivery trucks moving along the cell's highways. As for how they work, they walk along the microtubules using a cycle of binding, ATP hydrolysis, and changing their shape. Myosins serve a lot of important purposes: muscle contraction, cell division, cell movement, and even moving things inside cells along actin filaments. You could say they are the things that contract and power everything from bending your biceps to a cell moving across a surface. Here's how it works. Myosin heads attach to actin, then pull, let go, and reattach in a repeating cycle that uses ATP for energy. This makes the filaments slide past each other, which causes movement. Now, about the bacterial flagellum. Its main job is to push bacteria through liquids, allowing them to move toward or away from chemical signals, a process called chemotaxis. Think of it like a self-assembling, spinning outboard motor. Unlike the flagella found in more complex cells, the bacterial flagellum spins like a propeller. It gets its power from a flow of protons, or sometimes sodium ions, across the cell membrane. This powers a tiny motor built right into the membrane. The whole system is super complex, with a stator and rotor setup. ATP synthase is another cool rotary motor. Its main job is to make most of the ATP, the cell's main energy money, from ADP and inorganic phosphate. You can think of it like a tiny molecular turbine or a small rotary engine. It works like this. A flow of protons or ions across a membrane makes one part of the enzyme, called the F0 motor, spin. This spinning action causes another part, the F1 motor, to change shape, and that's what actually helps make ATP. It's truly a great example of a rotary motor found in nature. Now, let's talk about information processing and synthesis machines, which are absolutely central to the flow and expression of genetic information. Ribosomes are at the heart of this process. Their main purpose is protein synthesis, also known as translation. Basically, they read messenger RNA sequences and put amino acids together into specific protein chains. Think of them as a very fancy 3D printer that makes proteins based on genetic blueprints. Structurally, ribosomes are made of ribosomal RNA and proteins. They move along the mRNA, bringing together transfer RNAs that carry specific amino acids and help form peptide bonds between those amino acids to build proteins. DNA polymerases, well, their main job is to copy DNA, making new DNA strands based on a template. This is super important for cell division and passing on traits. You can think of them like a super accurate copy machine for genetic information. How they work is these enzymes move along the DNA template, adding matching building blocks to a new growing strand. And, they can also fix mistakes to keep errors down. Now, RNA polymerases, on the other hand, are in charge of changing DNA into RNA, like mRNA, tRNA, and rRNA, which is really the first step in how genes are used. If DNA polymerase is a copy machine, then RNA polymerase is more like a molecular transcriber or an information transfer expert. They work by unwinding a part of the DNA double helix and making an RNA strand that matches one of the DNA strands. Helicases. Well, their main job is to unwind double helices of DNA and RNA, separating the two strands. This function is super important for things like DNA replication, transcription, and repair. You can think of them like a molecular zipper. How they work is, they stick to the nucleic acid strands and, powered by ATP, they move along, breaking the hydrogen bonds between the bases. Protein folding and quality control machines are systems that make sure proteins get their correct 3D shapes, which is, absolutely key for them to work right. Molecular chaperones, like HSP70 and HSP60, also called chaperonins, are there to help newly made proteins fold correctly, stopping them from folding wrong or clumping together, and helping proteins that have been messed up by stress, like heat, refold. You could think of them as a protein's personal trainer or a molecular folding station. How they work is some chaperones stick to unfolded or partly folded proteins to stop them from clumping, while others, like chaperonins, make these barrel-shaped structures that trap the misfolded proteins and give them a safe place to refold, often using ATP to change their shape. So, proteasomes are these, specialized machines inside our cells. They have a super important job. They break down proteins that are folded, wrong, damaged, or just not needed anymore. This helps keep the quality of cell proteins in check. And, it keeps protein levels under control. You can think of a proteasome like a molecular shredder, or maybe even like a recycling plant. How they work is, these are big protein groups that find proteins marked for breakdown, called ubiquitinated proteins. Then, they unfold these proteins, and, they pull them into a central chamber, where they get broken down into smaller pieces, called peptides. Alright, let's talk about membrane transport and channels. These super cool machines control what ions and molecules can pass through cell membranes. For example, we have ion channels, like sodium channels, potassium channels, and aquaporins. Their main job is to make tiny holes in cell membranes. This lets specific ions or water molecules pass through. This is super important for things like nerve signals, muscles contracting, and even keeping cells the right size. You can think of them as very picky molecular gates or tunnels. And here's how they work. They often open and close when certain things happen, like changes in voltage or when signaling molecules attached to them. Plus, they only let certain things through based on their size and electrical charge. Now, let's move on to protein pumps, like the sodium-potassium ATPase and proton pumps. Their job is to actively move ions or molecules across membranes, even against their concentration gradient. And that process needs energy, usually from ATP. You could think of them like escalators or molecular pumps. The way they work is pretty clever. They change shape, which lets them grab onto the target molecule on one side of the membrane, use ATP, and then release the molecule on the other side. Next, we're going to dive into structural and maintenance machines. These are the ones that help with the cell's physical shape and how it holds together. Cytoskeletal filaments, like actin, microtubules, and intermediate filaments, are a great example. Their main job is to give structural support, let the cell change its shape, and act as pathways for molecular motors. Now, even though individual filaments aren't exactly machines in the same active way that motors are, how they come together and fall apart are carefully controlled processes that create these really dynamic structures. You can think of them as the scaffolding, roads, and internal skeleton of the cell. The way it works is through the building up and breaking down of protein parts like actin and tubulin. These are controlled processes that allow the cell to build, break down, and reshape its internal structure. So, to wrap things up, this list is by no means complete, because the complexity of cell machinery is truly amazing. Every day, researchers are finding new molecular machines and figuring out the tricky ways they work inside the cell's super-connected system. Thanks a lot for watching the video. If you liked it, please hit the like button, subscribe for more cool science stuff, and share it with your friends. We'd love to hear what you think and any questions you have, so leave a comment below and join the conversation.